Welcome to our podcast on muscle. This is going to be where we look at different images of skeletal, cardiac, and smooth muscle and learn to be able to distinguish them from one another as well as from other tissues. So we're going to start out, much like we do in all of our lectures, we're going to start out with skeletal muscle and look at those characteristics and be able to identify these tissues. So I think this is fairly straightforward even at this high magnification. As we look at this tissue, we see our alternating light and dark pattern. We can pretty easily see our I band, A band. We can even see the Z line right down the middle. And first, our eyes are drawn to these long cables, and at first it seems like this is going in and out of the plane of section, so you may be thinking, well, is this branching? Certainly we're going to see much more branching than that, but your eye probably is going to be able to be drawn toward the peripheral nuclei. There's our peripheral nuclei. I think this is a tangentially cut one. It's not in the middle. Might give you the impression of that. But the other reason that I'm calling this skeletal muscle is look across the muscle cells. Follow this I band. It's almost practically straight all the way down. Even over here, as we follow this across cells, you can see how really regularly aligned up they are. And so that regular alignment across muscle fibers is why I call this skeletal muscle, and is in fact skeletal muscle. Now that's longitudinally section. Go to cross section, again, even high magnification. We can see the connective tissue surrounding each one of these cells. And what do we call that? It's the endomysium. We can see our multiple nuclei, and they're all in fact peripheral. We have some capillaries in here as well. So this is a typical pattern of our skeletal muscle in cross section. Now, other things we're going to see in these cross sections, not necessarily in that same grouping as we saw here. We can see some, again, cross sections of skeletal muscle. We have the capillaries. Really are hard pressed to see the endomysium of these. Then we get to this connective tissue. It's outside of the muscle fibers themselves, and this is probably going to be our paramecium because we don't see a surface. Here's more muscle cells, and likely over here we've got some blood vessels going on here. But then all of a sudden we have this connective tissue and then what looks like muscle fibers inside this bundle of connective tissue, sort of isolated. These are our intrafusal muscle fibers, part of our muscle spindle apparatus. And here we have another over here as well. These are, again, the contractile fibers are extrafusal, and these are intrafusal. So this is our muscle spindle. Lower mag now, as we pull back away, we can see again this nice marble pattern that you typically see like when you buy a steak. And so if we look on the outside of the whole muscle organ, this connective tissue is called the epimysium. And as I mentioned, you also have a paramecium closer to the outside of this fascicle. Here we have one fascicle, but I'm never going to put the pointer here and say name this. If it's on the outside, I'm going to say epimysium. If I'm looking for paramecium, I will be between fascicles. And if I'm looking for endomysium that wraps each muscle cell, it's going to be a much higher magnification view, and it's going to be deep within one of these fascicles. So here, a little bit more close look. This is going to be our paramecium. We've got some fibroblastic cells running through here, collagen fibers. Between our muscle cells, this is going to be our endomysium. Got some capillaries in here. Again, notice the peripheral nuclei, and that will always lead you down the straight and narrow. You won't get this confused with either cardiac or smooth because those nuclei are centrally located. Now, I included these. These are just some very interesting cuts. These are kind of oblique, tangential, not along our typical transverse. and longitudinal that we're looking at, and some of these muscle fibers and bundles are running parallel to each other, and some are running perpendicular to each other. But it just makes for a really interesting looking and almost artistic section through skeletal muscle. This is not one you'll see on a quiz from me, but I just want to include it because I just thought it looked really cool. But it gives you a great appreciation for how these fibers can be cut in such different planes of section to make it very challenging to identify, especially at a low mag like this. But if you look at the modeled color, you can see some darker and some lighter. This is really getting into our fiber types. And so once again, won't ask you to identify these at the light level for the lab, but you will need to know those differences in fiber types. We studied them in physiology. You need to hang on to those. It's important that you know those. The red fiber types, the white fiber types, the fast and the slow muscle, because that's really what we're looking at here. The difference in the amount of glycogen, the differences in the amount of myoglobin that are present will give these each a different stain. Here you can see our type 1 fiber, our type 2A, and there they have 2B. I like 2X. That's what I've always learned those as. And so all these different types can be seen with various stains. So now shifting gears to the EM level, we can see the banding pattern that we've come to appreciate in the A band, the I band. There's our Z line. Right in the middle we have the pale H band and the dark M line right down the middle. Here we're sort of between, going out of the plane of section, we can see some sarcoplasmic reticulum. Got some elongated mitochondria in here. We can't really see T-tubules at this level, but we'll look at those as we go a little bit higher magnification. So once again, sarcoplasmic reticulum. On this terminal side, we're going to have terminal cisternae that's enlarged. There's a little section through a T-tubule that we'll see. Our mitochondria, A band, I band, Z line, H, and M. Once again, even higher, getting a better look at the I, and I don't like these quite as much, but again, we can begin to look in here and we can see some of our T-tubules, sarcoplasmic reticula. There's one of our triads, T-tubule in the middle, terminal cisternae on either side. There's another triad of 3 that we can see there. Sections through some mitochondria, other cut sections through sarcoplasmic reticulum. So banding patterns not nearly as interesting as what we're looking at, these high mags, as some of these other cellular components. All right, now looking at, again, some hollow organs. This is where we're going to find a couple of different muscle fiber types and muscle types. As we look at this, this is going to be a cross-section through esophagus. We will get to that when we study our digestive tract. But if you remember, the esophagus, at the very top third of the esophagus, that's primarily going to be skeletal muscle, where you initiate a swallow voluntarily. But then as you move lower and lower, you lower the proportion of skeletal muscle and increase the proportion of smooth muscle. So in this particular tissue, we're actually going to see 2 fiber types mixing and blending together. And in this micrograph, we can see that we have a cross-section through peripheral muscle. We got peripheral nuclei, so this is skeletal muscle. Here we have another skeletal muscle fiber, that's what all this is. But then all of a sudden we see these rounded cells, large central nucleus. So this is going to be our smooth muscle cut in cross section. So here you can sort of pick up the difference between a smooth muscle cell and a skeletal muscle cell. Again, looking at similar areas, different stain this time. Here we see our somewhat obliquely stained skeletal muscle. Here you can see the nice striations of our skeletal muscle peripheral nuclei. But then you get down into this area and we're getting into more of this oblique or maybe transverse running of our smooth muscle. So again, a nice comparison between striated that we see here and smooth muscle. Now we're shifting gears even more. As we look at this image, the first thing that you should be impressed with, not with just this material right in the middle, just ignore this for now, but you see tons and tons of nuclei. Now, you see the reddish pattern to this, the wavy pattern that you might see up in this portion of the image. It almost gives you the impression that we're looking at dense, regular connective tissue, but these nuclei are around. There are vastly so many more of these nuclei present. We don't really see a good banding or striated pattern. We might have a hint of it down here, but certainly these are interwoven and interconnected sheets of tissue. So what we're doing now is we are transitioning out of skeletal muscle, and what we're looking at is cardiac muscle. And in fact, this particular bundle of cells here, these are going to be our Purkinje fibers, our conductive cells within cardiac muscle. So when you see something like that, very nervous system tissue looking, I know we haven't got to the nervous system yet, but it's going to be rather light, pale staining tissue surrounded by all of these nuclei, flowing cells arranged into sheets. You're pretty much going to be thinking either smooth muscle or cardiac, and in this case, cardiac muscle would be the tissue. So we're going to look at cardiac muscle. Here we have some large sections of cardiac muscle. This is a low magnification view of atria and ventricle. And as we look closer at tissue, we are going to find those characteristics. We see the intercalated disc running between them. Here we see our striated pattern. It's going to be offset as these cells branch in and out of the plane of section. And here we have our large centrally located nucleus. But the intercalated disc by far is the easiest characteristic to pick up on and identify. Higher magnification again, we see the stair-step look to our cardiac muscle, centrally located nucleus. Sometimes you may see multiple nuclei. Here we see 2 nuclei in this particular frame. But again, the intercalated disc and the disjointed, misaligned A and I bands are going to be the indicative characteristic of cardiac muscle. So here in this micrograph, once again, as we're looking, we can automatically find our intercalated disc. That's easy, not just because of the inset, because we see that. But here you see how disjointed the sarcomeres are, the I band, the A band. Tons of mitochondria interspersed throughout here. That's going to be your tip-off that we're looking at cardiac muscle. It almost looks disorganized to me when you compare it to skeletal muscle. Once again, as we follow our sarcomeric pattern, we have our I band with the Z disc, A band, and as we approach the next I band, we no longer have a Z disc, but we have this very irregular, jagged stair step. This is our intercalated disc. Some of these are darker than others. Some of these are going to represent gap junctions. Areas such as this, where we're running almost parallel to the sarcomeres, this is going to be where we will have typically gap junctions, versus where we have the transverse portion that is perpendicular to the vertical forces here, to the forces of our contraction. That's where we're going to have desmosomes. But when it's all twisted and bent through here, it's difficult to really pick up the transverse versus the longitudinal portion that we draw out nice and regular in our textbooks. Now we're going to look at smooth muscle, and one of the places where we find smooth muscle are around some of our certainly larger blood vessels, but even some of the smaller ones. And as we look at these lamina surrounding our blood vessels, we're going to look at some large arteries And we have a nice stain that's going to be staining blue for our collagen or reticular fibers and red for smooth muscle. So we can see how the smooth muscle is going to be arranged circumferentially around these blood vessels. So when these smooth muscles contract, they're going to narrow or constrict the lumen of the blood vessel. Now we're looking in part of our intestinal tract. We have certainly our epithelium that lines our lumen, because remember, epithelia lines or covers. And then as we go deeper into this tissue, we're going to have some smooth muscle in a very thin band just beneath our epithelium. And then we're going to have a thick band of smooth muscle longitudinally arranged and circumferentially arranged out in this layer of our alimentary canal. And again, these names, mucosa, submucosa, externa, we'll get to those when we get to our organ histology. Right now, we're just looking at recognizing fiber types. Much like we talked about in lecture, here we have a great view of cross-section of smooth muscle versus longitudinal section of smooth muscle. Easy to pick up those centrally located nuclei, our endomysium or external lamina. It's the same terminology, means the same thing around these cells, versus our elongated oval-shaped centrally located nuclei in this streaming smooth muscle. Another location for smooth muscle is going to be our urinary tract. Here we're looking at urinary bladder. We have our epithelium lining this. And do you remember the epithelium of our bladder? What is that classification going to be? Transitional epithelium. We can't see it here, so I was just asking you for your location. And then as we get down into these deeper regions, we're going to see different layers of our smooth muscle. So once again, we have our transitional epithelium. We have just small bundles of circumferential smooth muscle, and then these larger sections of smooth muscle. All these nuclei present here, this reddish color, which is going to be indicative of muscle, it's going to tip you off that we're in a hollow organ. We're looking in these deeper layers. That's going to be our smooth muscle. Again, different view of the same thing, bundles and bundles and bundles of smooth muscle. We can sort of see some transverse sections of our smooth muscle in this location, and then we've got more of a longitudinal section through here. You can almost see that flowing or streaming view, and then different twisted up views here. It's difficult to pick those out. Here's some more longitudinal sections of smooth muscle right there. Now at the EM level, once again, we're going to see cross sections, nice centrally located nucleus, fairly round cell. In these cross sections, it's difficult to really pick up what's going on, but as we look at a more longitudinal section through our smooth muscle, all these little black spots, those are going to be the dense bodies. Those are the attachment points for our actin and myosin myofilaments inside that smooth muscle cell. Higher magnification again, showing these dense bodies. The little black spots look like pepper scattered throughout the plasma membrane of our smooth muscle cells. Now, even higher magnification of our cells, we can see myofibrils running through here, our actin and myosin thin filaments. We've got some mitochondria, and then these little spots that we see right here. I believe these are going to be some of the caveolae in which we store our calcium to be released so that we can get a contraction.